Yes, so I'm Marcus Dorner. I've been working at Imperial College London for the past 4 years as a non-clinical senior lecturer. And before that, I've been in the States working in New York on viral hepatitis. So really, I've been working in the field of viral hepatitis for the past 8 years. And before that, I was in Switzerland working on my PhD. That was actually during my graduate research studies in Munich. So originally I'm a trained chemist, and during my master's thesis I had a project that was purely based on protein characterization, and there I learned how the host, so humans, actually developed a lot of pattern recognition mechanisms, so how the host actually recognizes infections. But what triggered for me the main interest was that the cells can actually identify that there is virus DNA, for example, coming into the cell. So how the host actually distinguishes the viral DNA from itself, its own DNA, is something that quite struck me. And then from there on out, I decided, okay, virology is really a field I wanted to move into. So I moved to Switzerland for my PhD on herpes viruses, and there was really sort of like the spark of interest for virology, chronic viral infections, how they evolved over the millennia to actually subvert the host completely in some instances. And then following that, I decided I wanted to work on viruses that are still chronic viral infections, but that are more pathogenic to humans, that are more dangerous and causing more of disease to humans. So I moved on to work on hepatitis C virus, where sort of like over the course of 4 years we saw really this enormous development of drugs that are now in clinic that brought hepatitis C virus from a disease that is virtually curable in only about 30 to 40% of cases to now about 95 to 100% of cases who get cured with a simple 12 to 24 week drug treatment. So moving from there, I then decided, well, I want to become independent as an own academic investigator, and moved here to Imperial College London in order to study mostly chronic viral infections. So at the moment we're looking into hepatitis B, hepatitis C, and also an HIV infection. What really struck me as the key factor that limits research to a large degree is the model systems that are available to study these diseases. So what we've done in the past already is work on creating small animal models for hepatitis C virus. At the moment we're more focused on hepatitis B virus, creating small animal models for hepatitis B virus, and also in 3D bioreactor systems. So we have microfluidic culture systems that actually allow you to study a lot of the aspects that previously could only be studied in let's say chimpanzees in the past, or in human patients infected with the virus. We can now study in vitro. So what we're currently looking at is how these viruses actually manage to be chronic in humans, so lifelong persist in a human, without actually being recognized or fought by the immune system to a large degree. So what actually did the virus evolve to subvert immune responses in humans? Yes, so it has actually showed us an insight into which parts of the immune system in, like, for example, the mouse model, is able to control hepatitis C virus. So we've done some very detailed characterization into which parts of especially the innate immune system, are responsible for allowing hepatitis C virus to replicate in human cells, and which parts restrict hepatitis C virus from actually infecting mice. In addition to that, we're now involved in some vaccine trials that actually could become important for actually introducing novel hepatitis C virus vaccines. So the main challenge would be that the virus actually, or the host never develops immunity to hepatitis C virus infection by itself. So if you're infected and you get cured, you can still become reinfected with the virus in the future, and you will not be able to defend yourself from infection. Developing a vaccine that is actually causing the immune system to recognize all future incoming hepatitis C virus is going to be very difficult. And in addition to that, the virus itself is very heterogeneous, so the virus mutates very rapidly, and a lot of the, for example, antibodies that are created in order to prevent future infections may not be effective against a virus that has mutations. So that might be the biggest challenge. Yes, so hepatitis C definitely is an immediate candidate for eradication with novel treatments are now available. So you can actually cure the majority of people that are infected at the moment. For hepatitis B virus, it is definitely a disease that can be eradicated. However, the problem is that there is no cure for hepatitis B virus. The major issues in hepatitis C virus treatment at the moment is drug availability. So prices for novel hepatitis C drugs are still astronomical, so a lot of countries, especially in developing countries, do not have access to treatment because of economic reasons. This is definitely something that would be an urgent need to address in order to eradicate hepatitis C virus. For hepatitis B virus, definitely enhancing access to the vaccine, making sure that developing countries have better access to hepatitis B vaccinations at birth, can prevent lifelong infection of people. But those two diseases, especially since they only infect humans, are a prime candidate for eradication because you don't have to worry about an animal reservoir that spreads the virus around. One of the major hurdles to overcome in curing the world of hepatitis C virus, for example, is that infection usually goes unnoticed. So the majority or a large fraction of people that are infected don't know that they are infected. So actually getting awareness to people that they should get tested so that they can get treated and don't spread the disease is essential for that. In addition to that, with all the new hepatitis C virus drugs that are now on the market, it's obviously important to raise awareness for availability of these drugs and raise awareness of the fact that it now became a curable disease, but also remind people that research into hepatitis C virus is still needed because we don't understand every aspect of the infection yet. So I would envision that there will be a hepatitis C virus vaccine in the future. So that would definitely be something that would be quite nice to introduce in the world as an alternative to treatments that are available. In addition to that, I think at the moment in hepatitis C virus, the problem shifts more to a public health problem where identification of infected patient populations and the cure of those patient populations will become extremely important in the coming years. In hepatitis B virus, it's actually much more complicated because very much less is known about the infection itself, and the model systems are still very suboptimal for studying hepatitis B virus effectively. So it will take a significant longer period of time for effective treatments to become available for hepatitis B virus. However, we hope to contribute to basically providing better model systems to the academic community so that we can actually start working on curative treatments for hepatitis B virus as well.